Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding the basic differences in between mitosis and meiosis from cell cycle and cell division in detail. So at first, if we consider mitosis, so here what is mitosis? Mitosis, it is a type of cell division which is observed in case of somatic cells or vegetative cells. But whereas meiosis, it is a type of cell division which is observed in case of reproductive cells or in case of sex cells. Is it clear? So let me write it on the board. Mitosis. Mitosis is a type of cell division which is observed in case of somatic cells or vegetative cells. But whereas meiosis, it is a type of cell division which is observed in case of reproductive cells or in case of sex cells. Is it clear? Meiosis is a type of cell division which is observed in case of reproductive cells or sex cells. So here most commonly mitosis, it is a type of cell division which is observed both in case of haploid cells and diploid cells. But whereas meiosis, it is observed only in case of diploid cells but not in case of haploid cells. Is it clear? Mitosis is observed both in case of haploid and diploid cells. But whereas meiosis, it is observed only in case of diploid cells but not in case of haploid cells. Meiosis is observed only in case of diploid cells but not in haploid cells. So here most commonly mitosis, it is termed as, or it is otherwise known as, equational division. So here why mitosis, or it is otherwise known as equational division? Because the specific cell division, it is responsible for the maintenance of constant chromosomal number both in case of the parental cell and even in case of the daughter cell. That means if the parental cell, if it has certain number of chromosomes, the same number of chromosomes they are observed in case of the daughter cell. So the reason why mitotic cell division, or it is otherwise known as equational division, but whereas meiotic cell division, or it is otherwise known as reductional division. So here why the specific meiotic cell division, or it is otherwise known as reductional division here? Because the specific meiotic cell division, it is responsible for reducing the chromosomal number exactly to half. So the reason why meiotic cell division, or it is otherwise known as reductional phase or reductional division. So let me write it here. Mitosis is otherwise known as equational division because it maintains constant chromosomal number. Constant chromosomal number in parental cell. In parental cell and daughter cell. In parental cell and daughter cell. But whereas meiotic cell division, or it is otherwise known as reductional phase or reductional division. Meiotic cell division. Meiotic cell division is otherwise known as reductional phase. Reductional phase or reductional division. Reductional division. And here, in case of the mitotic cell division, the specific parental cell, it produces two identical daughter cells. But whereas in case of the meiotic cell division, the specific parental cell, it produces four haploid daughter cells. Is it clear? So let me write it here. In case of mitosis, the specific parental cell produces two identical daughter cells. But whereas in case of meiosis, four haploid daughter cells are produced. Four haploid daughter cells are produced. So let me arrange the things here. And then after, the next basic difference in between mitosis and meiosis, it is observed in case of regulation. So here mitosis, it is most commonly it is responsible for the regulation of cell growth, healing and DNA repair. But whereas meiotic cell division, it is responsible only for the regulation of sexual mode of reproduction. So let me write it here. Mitotic cell division is responsible for the regulation of cell growth, healing and DNA repair. But whereas meiotic cell division, it is responsible only for the regulation of sexual mode of reproduction. Meiosis is only responsible for the regulation of sexual mode of reproduction. And then after we will be studying regarding the basic differences in between each individual phase in between mitosis and meiosis in detail. So at first, if we consider the prophase of mitotic cell division, so here the prophase of the mitotic cell division, it remains very simpler, but whereas the prophase of the meiotic cell division, it remains much more complex. So let me write it here. Prophase of mitotic cell division. So here the prophase of mitotic cell division remains very simpler, remains very simpler. But whereas the prophase of the meiotic cell division, it remains much more complex. Prophase of meiotic cell division remains much more complex. And here, in case of the prophase of the mitotic cell division, in general, we won't observe the bouquet stage and even the synapses. But whereas in case of the prophase of the meiotic cell division, in general, we observe the bouquet stage and even the synaptic stage. Is it clear? Bouquet stage and synapsis is not observed in prophase of mitosis. But whereas in case of the prophase of meiosis, in general we observe the bouquet stage and even the synaptic stage. Bouquet stage and synapsis is observed in case of prophase of meiosis. So here, what is bouquet stage and what is synapsis? In order to get the proper idea regarding the bouquet stage and the synapsis, you can watch my previous video sessions. Is it clear? And here, in case of the prophase of mitosis, in general, chiasmata and the crossing over formation does not take place. Chiasmata and crossing over formation does not take place. But whereas in case of the prophase of meiosis, in general, chiasmata and crossing over formation takes place. Chiasmata and crossing over formation takes place. So here all these are the basic differences in between the prophase of mitosis and the prophase of meiosis. And then after we will be studying regarding the basic differences in between the metaphase stage of mitosis and the metaphase stage of meiosis in detail. So let me arrange the things here. And if you look at the basic differences in between the metaphase stage of mitosis and the metaphase stage of meiosis in detail. So at first, if we consider the metaphase stage of mitosis, so here basically during the metaphase stage of the mitosis, the specific chromosomes which generally do exist, they are termed as monovalent chromosome or dyad chromosome. But whereas the specific chromosomes which generally do exist during the metaphase stage of meiosis, here they are termed as bivalent chromosomes or tetrad chromosomes. So let me write it here. During the metaphase stage, the specific chromosomes which generally do exist are termed as monovalent or dyad chromosomes. But whereas in case of meiosis, during the metaphase stage, the specific chromosomes which generally do exist, here they are termed as bivalent or tetrad chromosomes. During the metaphase stage of meiosis, the specific chromosomes which generally do exist are termed as bivalent or tetrad chromosomes. Bivalent or tetrad chromosomes. So here, in case of the metaphase stage of mitosis, in case of the monovalent chromosome, within the monovalent chromosome, here we can observe two sister chromatids. But whereas in case of the metaphase stage of meiosis, within the bivalent chromosome, here we can observe four sister chromatids. Monovalent chromosome has two sister chromatids. Is it clear? So just look at here. So here it is nothing but what? Monovalent chromosome. So here the monovalent chromosome or the dyad chromosome, basically it has two sister chromatids. But whereas in case of the metaphase stage of meiosis, the specific bivalent or the tetrad chromosome, it has four sister chromatids. Specific bivalent or tetrad chromosome has four sister chromatids. And then after, in case of the metaphase stage of mitosis, in general, the specific monovalent chromosomes, they are capable to remain in association with the spindle fibers by taking the advantage of a specific disc-shaped protein complex termed as kinetochore. Is it clear? The specific monovalent chromosome or the dyad chromosome, it is capable to remain in association with the spindle fibers by taking the advantage of a specific disc-shaped protein complex termed as kinetochore. So here, by taking the advantage of a disc-shaped protein complex termed as kinetochore, here the chromosomes, they are capable to remain in association with the spindle fibers. So here the specific region where the chromosomes, they are capable to align with the spindle fibers during the metaphase stage, here it is termed as equatorial region or metaphase plate. So here, in case of the equatorial region or the metaphase plate, in general, the centromere, the specific centromere, in general, it remains above the equatorial region, but whereas the sister chromatids, in general, they are exposed towards the spindle poles. Is it clear? Within the equatorial region or within the metaphase plate, the specific centromere, in general, it remains above the equatorial region, but whereas the sister chromatids, in general, they are exposed towards the spindle poles. So let me write it here. During the metaphase stage, within the equatorial region, centromere remains above. Centromere remains above the equator, but whereas sister chromatids are exposed are exposed towards the spindle poles. Towards the spindle poles. Is it clear? But whereas in case of the metaphase stage of meiosis, in general, as we all know, within the metaphase stage of meiosis, the specific chromosomes which generally do exist, here they are termed as bivalent or tetrad chromosomes. So here, because of the presence of bivalent or tetrad chromosomes, here genetic exchange takes place or crossing over takes place. So here during the genetic exchange or the crossing over in between the non-sister chromatids of homologous chromosomes, in general, the centromere, it exposes towards the spindle poles. Is it clear? During the genetic exchange, or crossing over during the prophase, in general, centromere exposes towards the spindle poles. Centromere exposes towards the spindle poles. Is it clear? And then after we will be studying regarding the basic differences in between the anaphase stage of mitosis and the anaphase stage of meiosis in detail. So let me arrange the things here. And during the anaphase stage of mitosis and meiosis, so here during the anaphase stage of mitosis, in general, the specific monovalent chromosome or dyad chromosome which has two sister chromatids, they are ready to undergo separation. So here the two sister chromatids, they are ready to undergo separation because of the splitting of centromere. So here the splitting of centromere, it occurs only in case of the anaphase stage of mitosis, but not in case of the anaphase stage of meiosis. In case of the anaphase stage of the meiosis, splitting of centromere does not take place. Here, in case of the anaphase stage of meiosis, segregation of homologous chromosomes takes place. That means after the successful regulation of genetic exchange or crossing over in between the non-sister chromatids of homologous chromosomes, now here these homologous chromosomes, they are ready to undergo separation during the anaphase stage of meiosis. So let me write it here. During the anaphase stage of mitosis, separation of sister chromatid takes place. Separation of sister chromatid takes place because of the splitting of centromere. Because of the splitting of centromere. But whereas in case of the anaphase stage of meiosis, in general, splitting of centromere does not take place. Here in case of the anaphase stage of meiosis, here only segregation of homologous chromosomes takes place. In anaphase stage of meiosis, in general, separation of sister chromatids, does not take place. Because of the absence of splitting of centromere. So here, once when the splitting of centromere, once when it occurs in case of the anaphase stage of mitosis, now here the specific sister chromatids, they are ready to migrate towards the spindle poles. And here, these sister chromatids, they act as future chromosomes in the upcoming daughter nuclei. Is it clear? So let me write it here. The specific sister chromatids which undergo separation they are ready to reach the spindle poles and they act as future chromosomes in the upcoming daughter nuclei. Future chromosomes in the upcoming daughter nuclei. And here, in case of the anaphase stage of meiosis, during the anaphase stage of meiosis, only segregation of homologous chromosomes takes place. Segregation of homologous chromosomes takes place after the regulation of genetic exchange. Is it clear? And then after we will be studying regarding the basic differences in between the telophase stage of mitosis and the telophase stage of meiosis in detail. So let me arrange the things here. And during the telophase stage of mitosis, so here during the telophase stage of mitosis in general, the specific nucleoli which successfully disappears at the end of prophase, again it reappears during the telophase stage. And here once when the splitting of the centromere takes place during the anaphase stage, here both the sister chromatids initially which are under associated form, they are ready to undergo separation. And here each sister chromatid, it acts as a daughter chromosome in the upcoming daughter nuclei. But whereas in case of the telophase 1 of the meiosis 1, so here in case of the telophase 1 of the meiosis 1, likewise the telophase stage of mitosis, again the specific nucleoli, it successfully reappears. But here we need to understand that within the telophase 1 of the meiosis 1, instead of having one single sister chromatid as a chromosome, here we can observe a dyad of chromosome or monovalent chromosome, because in case of the anaphase 1 of the meiosis 1, splitting of the centromere does not take place. Because of the absence of the splitting of the centromere, here sister chromatid does not undergo separation. Instead of undergoing the separation of sister chromatids, here the segregation of homologous chromosomes takes place. So the reason why at the end of the telophase 1 of the meiosis 1, here we can observe a dyad of chromosomes. Is it clear? So let me write it here. During the telophase stage of mitosis, the nucleoli successfully reappears. And the sister chromatids, the specific sister chromatids which undergo separation act as chromosomes chromosomes in the upcoming daughter nuclei. In the upcoming daughter nuclei. Daughter nuclei, right? And here, in case of the telophase 1 of the meiosis 1, nucleoli, again it reappears. During the telophase 1. And instead of having sister chromatid as chromosome, here dyad of chromosomes are present. Is it clear? So here in case of the telophase of the mitosis, here single sister chromatid, it acts as a chromosome. But whereas in case of the telophase 1 of the meiosis 1, here we can observe a dyad of chromosome. So here it is nothing but what? Dyad of chromosome, right? Because in case of the anaphase 1 of the meiosis 1, splitting of the centromere does not take place. So the reason why at the end of the telophase 1 of the meiosis 1, here we can observe a dyad of chromosome. And here, within the telophase stage of the mitosis, in general, spindle fibers, they undergo complete disappearance. And during the telophase 1, of meiosis 1, spindle fibers does not undergo complete disappearance. Complete disappearance, right? And here the genetic content within the parental cell and daughter cell, it remains very similar in case of the telophase stage of mitosis. But whereas in case of the telophase 1 of the meiosis 1, the genetic content within the parental cell and the daughter cell, it won't remain similar. Genetic content remains similar both in the parental cell and daughter cell. But whereas in case of the telophase 1 of the meiosis 1, genetic content does not remain similar within the parental cell and daughter cell. Within the parental and daughter cell. Right? So here, all these are the basic differences in between mitosis and meiosis in detail. So here in the next upcoming lecture session, we will be studying regarding the basic differences in between meiosis 1 and meiosis 2. So here I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.